Hello. Okay, today we are in this next video, we're going to talk about how um, the nucleotides that we talked about in the previous section are linked one after another in a single strand. So that's going to be the goal of this lesson. So let's just break this question or this objective apart a little bit. So again, like DNA that we know, I'm going to make that a little bit bigger. DNA, as we know, is a double strand, right? Let's see if I can copy over this. A double strand of these. So if you can picture that these four nucleotides joined together would make up one of these strings or one of these strands of DNA. And then I'm sure you've seen in pictures where we have like the little ladder kind of shape that represents it. We'll talk about that ladder part in a little bit. So this assessment statement is just saying, like, how do we talk about how these one strand of DNA is joined together? And it's not so complicated. So I'm going to draw it, and we'll talk our way through it. So I'm going to draw two of these. So always good practice to draw nucleotides. So that circle represents what? Our phosphate. And then we have a pentagon that represents our sugar. For DNA, the sugar is deoxyribose. And then this rectangle here represents our nitrogenous base, which can be any of these four: adenine, cytosine, thymine, or guanine. Now, one of the things that you need to remember is that if you remember when we were talking about the structure of ribose, if we looked at these individual atoms, at the top here, there was a molecule of oxygen. And then at each one of these little corners, we have a carbon here, we have a carbon at this corner, we have a carbon at this corner, we have a carbon at this corner, and then there's another carbon like located right here. So let's erase that, and we can write a little carbon there. And there's a carbon here that's connected to this carbon, and then eventually to our phosphate molecule. Now, these carbons are numbered. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And that's kind of important. So when you're drawing and labeling, when it asks you to do that, it's very important that our nitrogenous base has a covalent bond to carbon number 1. And then carbon number 2, carbon number 3, carbon number 4, and very important that carbon number 5 is connected to our phosphate here. You do not have to add the numbers when you're drawing and labeling, but it's very important that you at least put them in the right parts of this pentagon. And when we start talking about other things that DNA can do, like replication, and we start talking about how this molecule eventually makes a protein, then these carbon numbers are going to be actually important. Okay, get the eraser out again because we have to draw maybe a couple of them linked together in a strand. So let's draw another one. So we have a covalent bond here coming off which carbon? Carbon 1, carbon 2, carbon 3. That's going to be connected to the phosphate of our second nucleotide. And that will connect to carbon number 5. Then we have another deoxyribose, and the nitrogenous base covalently bonded to our deoxyribose, right? So that's how this works. That's how they're linked together. Carbon number 5 to the phosphate in the same nucleotide, and then carbon number 3 to the phosphate of different nucleotides, because this is a different nucleotide than this. And now we're growing our DNA. Let's do one more. There's a covalent bond off carbon 3 of nucleotide number 2. Why don't we just call this number 2 for reference? So off nucleotide number 2, connecting to the phosphate of our third nucleotide. There's our pentose sugar connected to our nitrogenous base. A couple things about this. We will quite often call the phosphate and the sugar part, so the phosphate and the deoxyribose part, we're going to call that the backbone of our DNA. So this dark line here that I have is repeating phosphate, sugar, phosphate, sugar, phosphate, sugar. And these little lines in between them are our nitrogenous bases. Okay, so that's how you can talk about how they're linked together. Let's talk about multiple strands now. So that is the next slide. So on 2.64, explain how the DNA double helix is formed. And when we say double helix, we're talking about two strands creating this twisting staircase or twisting ladder made of these things called complementary base pairs. So I'm going to erase this mess here. So a couple things first. So when we're talking about complementary base pairs, we're talking about bases that complement each other. And it's not like, hey, you look great today. No, we're talking about they match. So this type of complement with an E means that they're matching. And in the world of nucleotides, DNA, A will always complement T. So adenine and thymine always go together, and guanine and cytosine always go together. And the reason is the number of hydrogen bonds. So now we're talking hydrogen bonds between nitrogenous bases of different strands of DNA. So if you look at cytosine and guanine, there are 3 hydrogen bonds. And if we look at adenine and thymine, there are 1, 2 hydrogen bonds. So it's actually very important that you draw the correct numbers in between those bases. So let's do one real quick. So I'm going to draw just 2 of them stuck together just so you can kind of see how it looks. Again, the phosphate, the deoxyribose represented by a pentagon. Off of carbon number 1, we have our nitrogenous base. And then I'm going to draw another one on this side. Now, if you notice, they look different, right? If you look at this pentose sugar and you look at this pentose sugar, they're not facing the same direction. This point is pointing up, and the point of this one, right, is pointing down. We call this anti-parallel. They're going in opposite directions, and it's very important that you draw them that way. So I'm going to draw hydrogen bonds between the nitrogenous bases. Here's another nitrogenous base here. Covalent bond to carbon number 1 of my upside down pentose sugar. And then my phosphate will be off carbon number 5, but it will be pointing down also, right? Because this is 1 nucleotide. And that's how you draw it. So just, can you tell me, is this nitrogenous base, is it adenine or is it guanine, based on the information that I've given you? And your answer should be adenine, because it has 1, 2 hydrogen bonds, and adenine will always form 2 hydrogen bonds with thymine. And guanine will form 3, so it can't be guanine. All right, thanks.